Hello friends, so in today's lecture, we are going to talk about the another experiment which is carried out by Avery, macCarty and Macleod in 1944, which is also included into experiments in which the DNA is genetic material has proved. So we told about the first experiment, which is the Griffith experiment, which is also uploaded in my channel. You can watch it. Now the second experiment is Avery, macCarty and Macleod. So by the first experiment, by the Griffith experiment, it is concluded that transformation is occurred. Some genetic material, some any constituent which transfer from one cell to another cell to give a characteristic or genotypic and phenotypic characteristic. But which thing is transferred, it do not told in the Griffith experiment. So this was told by these three scientists. Now what they do, they use killed extract filter. Yani first they kill the extract and then they filter it out. So now in the liquid filtrate, the cell constituents were there like protein, carbohydrate, RNA and DNA. Now he treated this filtrate first with lipase. Now after treatment, they mix S strain and the enzyme treated S strain and R strain. Now after that, they give some incubation time. After that, they saw the transformation was occurred. So it means lipid is not a material which is transferred from S strain to R strain. Now the second thing we do, they treat it with protease enzyme and they inject into the S strain to see what happened. Now after the protease treatment, the R strain also become S strain, so transformation occurs. Again, so the protein is not a genetic material. Is it also concluded? Now this strain is further treated with RNase enzyme and injected into the S strain. Now after incubation, it is also transformed. So transformation is occurred. So the three things, lipid, protein and RNA, it is not a genetic material, it is concluded. Now the third is a carbohydrate. Now it treated with carbohydrate and after treatment it injected the enzyme treated S strain with R strain, the transformation again occurred. And after at last they treated with DNase enzyme. And after the treatment with DNase and injected into S strain, after incubation, no transformation was occurred. So no transformation was occurred. It is due to the DNase enzyme is degrade the DNA present into filtrate and that side do not enter into the S strain and R strain is R strain become no S strain. So that's why the transformation wasn't occurred. So this is experiment told that DNA is a genetic material which transformed by these three scientists. I hope you understand. Thank you.